Good morning. I'm Souhei Sakata from Osaka Medical and Pharmaceutical University. I thank the organizers in Sofian Biosciences for giving me such a nice opportunity to talk about my study in such a nice symposium. I will talk about our recent study on zebrafish. Zebrafish lacking the Nav1.4, which is specifically expressed in muscle. We use zebrafish. Zebrafish are small fish, and the length of body is around 2 or 3 centimeters. This is adult, and we also use our experiment. We also use rubber in our experiment. The length is about 3 or 5 millimeter long, and they are transparent. This is one advantage of zebrafish. Transparency enable live imaging of cells and tissues by introducing fluorescence tag. This is an example expressing tag in muscle. Zebrafish also have other advantages. A high number of spin is of maintenance, and we can also generate the disease of human model, and gene is fully sequenced, and we can easily manipulate of genome to generate transgenic line and knockout line, knock-in line also. We start with the muscle contraction mechanism. I believe many of you may know well about the mechanism, but I will show you for someone who may not familiar with muscle contraction mechanism. And motor neuron innervates muscle, and acetylcholine is released from the motor neuron terminal, and it opens the gate of acetylcholine receptor, and membrane is depolarized. It is called end plate potential. End plate potential stimulates Nav1.4. This is the target of this my talk, and action potential is generated, and it stimulates dihydropin receptor, DHPR. It opens the gate of the ryanodine receptor on sarcoplasmic reticulum, and calcium ions are released from the SR2 cytoplasm, and muscle contracts. So Nav1.4 mediates new motor neuron excitability and muscle contraction. Zebrafish are vertebrates as well as human. So, but human fish muscle appears to be the same to the human muscle, but not it is always the case. And muscles are roughly divided into slow and fast fiber. And motor neuron innervates muscle, and motor neuron connect to at the center of the muscle tissue. That means the synapse of motor neuron located at the center of in both slow and fast fiber in human case. But zebrafish is not. The distribution of synapse is distinct between fast fiber and slow fiber. In slow fiber, synapse is located only on the both sides of the fiber. In fast fiber, the synapses are scattered all over the fiber. Not only the distribution, but also the orientation is distinct between two fibers. For slow fiber, the orientation is parallel to the body axis. Body axis means head and tail, this axis. For fast fiber, the orientation is not parallel to the body axis. One previous report recorded the sodium current from these fibers by wholesale patch clamp to identify fibers. The green dye was included in the pipette solution. So this is slow. This is fast fiber. And this is sodium current from fast fiber. And this is low current, and I recover the clearly voltage currents from voltage-gated sodium channel. But surprisingly, no sodium current in slow fiber in zebrafish. So we confirmed this result. No sodium channel expressed in slow fiber in zebrafish. So this raised many questions. One simple question is that the slow fiber can contract without sodium channel. How about fast fiber? Can fast fiber contract without sodium channel? I know this idea disagrees with the conventional view of mechanism of muscle contraction, but we tried. We generated Nav1.4 knockout fish. Zebrafish encode two Nav4 genes, SCN4A and B. We generated the at first single knockout fish. This is SN4A knockout fish, and this is SCN4AB knockout fish. And we cross these fish, and finally we generate a double knockout zebrafish. Then we analyze it. First, we estimate the swimming capability of zebrafish. Here is zebrafish rubber. Yeah, we tap the zebrafish to surprise. It's escaped like this. How about knockout fish? Fish is here. Surprisingly, the fish escaped with shaking the head side by side. The neck angle is plotted against the time. Plotted against time. We see no difference between two, and maximum turn angle also estimated, but we don't find any difference between two. To confirm that if knockout fish lose the sodium current, I performed the patch clamp recording from the isolated muscle fiber. This is a result from wild type fiber. We see clear inward sodium current like this. In addition to, if we add the TTX, the inward current was gone and outward current emerged. This outward current and inward current were recorded to be voltage-gated these currents are reported to be a current derived from voltage sensor movement of the hydrogen receptor, so-called gating current of DHPR. How about knockout fish? Even in the absence of TTX, we recorded this current. This is similar to this one. So this means sodium current completely gone in the knockout fish. So sodium channels is not dispensable for, if sodium channel is not dispensable for muscle contraction, wild type muscle also should contract even in the presence of tetrodotoxin, TTX. So we tried this idea. By calcium imaging, we generated a transgenic zebrafish line expressing GCAM calcium indicator specifically in fast muscle. And we isolated fast muscle and applied acetylcholine. And we see an increase of fluorescence. This is the result movie. We applied acetylcholine from right side. We see a clear increase of fluorescence. So, and in the presence of TTX, we applied acetylcholine from right side, and so I applied manually. So acetylcholine diffused, and fluorescence intensity increase like this. So given this result, we had no choice but to conclude that NAV is not always necessary for muscle contraction. Why can zebrafish muscle contract without NAV? One simple idea is end plate potential can directly activate DHPR, but in general, membrane potential is attenuated when traveling across the membrane. When it reaches to the DHPR, the magnitude of depolarization much smaller. And the end plate potential is originally is not large to stimulate DHPR. So focusing to this point, attenuation and depolarization amplitude, we performed mathematical stimulation whether the end plate potential can directly stimulate DHPR. The simulation is performed using a neuron software. This software is used for calculation of amplitude of depolarization or depolarization for propagation on axon, neural axon. So this simulator has a model of this cylinder, and in this simulator, axon is modeled as this cylinder. So we use this. We use this cylinder as a single myofiber, and depolarization and attenuation is calculated by the cable theory. Here, we estimated first attenuation. Here is a microfiber, and this gray shows a synaptic input, synaptic site. So synaptic current is injected at this site, and the membrane is depolarized, and the depolarization is depolarization propagates along this fiber. We calculated the depolarization at 0.L, 0.5L, 0.9L respectively. Here's the result. When L is 100 micrometer, and the depolarization of at each point is almost same, completely superimposed. That means no attenuation. When L is set to 200 micrometer, result is same. All results are completely superimposed. However, L is set to 500. We see a little bit attenuation. The black means 0.1, red and green is smaller than the amplitude of depolarization. When L is 100 micrometer, we see a clear attenuation. This is a picture of this is a picture of fibers from zebrafish rubber. The length is about, the length is not uniform actually, but the length is about 100 or 200 or so. So in this range, the simulation says no attenuation. So depolarization are not significantly attenuated in double muscle fiber. So amplitude, we estimate amplitude. For estimation to estimate amplitude, we need the number of synaptic site and amplitude input synaptic input amplitude. And input amplitude is estimated by the recording of miniature end plate current. And we recorded the current, and it is estimated to be 1.2 nanoamp on average. And here is a picture of the fibers, and this green spot means the synaptic sites. We counted the number of synaptic sites per one fiber. We found it 13 synaptic sites, and we inserted this cylinder shown in gray. And as shown here, the synaptic site is this distribution pattern of the synaptic site is not uniform among fibers. So I incorporated this gray area in equal interval. And we postulated the 1.2 nanoamp current are simultaneously injected at all synapses. And we calculated the membrane potential. The membrane potential is over 0 millivolt, suggesting that depolarization amplitude was large, sufficient for stimulation of the HPR. Summary of first topic, first part, we generated knockout fish, and we confirmed the knockout of no stream current in knockout fiber, but swimming capability is same to wild fish, and even the presence of TTX, wild fish fiber can contract. Simulation suggests that depolarization is not, attenuation is not significant, and the amplitude is also enough for stimulation of the HPR. So that means end-plate currents can directly activate the HPR. So this raises the next question. How about long fiber? This simulation suggests that if long fiber is short, attenuation can be avoidable. How about longer fish? How about longer fiber? How about the adult fish having long fiber? I address this issue next. Here's adult fish. I pinch the fin to supply the fish. And fish was surprised and escaped. How about knockout? Knockout also escaped like this. We found no difference between two, and turn speed also no difference. And we estimated swimming speed using swimming treadmill. And the flow is generated by the rotation of propeller, and the flow rate was increasing in a time-dependent manner. This is a video experiment. This is long, but I only show the first part and the end part, which begin to swim. And this is the end of the experiment. Fish swim as hard as they can, but give up swimming. The speed, the flow rate that fish give up swimming was defined as the maximum swimming speed. The maximum swimming speed is not is same between two. So I go back to simulation. Simulation of attenuation. Here's a picture of the fiber of adult fish. Adult fish, length of adult fish is about 100 micrometer. The simulation says membrane potential is attenuated in adult fiber. However, the synaptic sites are scattered. So simulation said that depolarization spread 100 or 200 micrometer is a radius of 100 or 200 micrometer around the synaptic site. So if this scattered synaptic site can cover all of the fiber may contract with a sodium channel, I suppose. So to test this hypothesis, we have learned, we performed simulation. for simulation, we need a number of synaptic sites and amplitude input currents. But unfortunately, we are not able to count the number of synaptic sites per fiber. As you can see, the the synaptic sites were labeled as line, not dots, dot, not dot. So we cannot count the number. And the currents, we cannot record the current because this fiber is very large to clamp the voltage by conventional patch clamp system. So I perform simulation by changing the number of synaptic sites, and we use the amplitude of 1.2 nanoamp. This is used for rubber simulation. So synaptic site was 11. The depolarization amplitude is very small. However, it was 51, the amplitude was bigger. And 101, and 139, we found huge depolarization over 0 millivolt. This means that scattered distribution of synaptic site and a high number of synaptic site enables the muscle to contract with us NAV 1.4. To summarize my talk, to second part, swimming capability of adult knockout fish was comparable to wild fish as well as larvae. The amplitude depolarization was attenuated significantly in the 100 micrometer long fibers. So it means in adult fiber. The simulation suggests that high number of synaptic sites in the scattered distribution of synaptic sites allow for normal contraction with that sodium channel 1.4. The need for muscle action potential is not universal, is a message from the Zen. This is acknowledgement. This study done in Professor Fumihito on lab in Osaka Medical Pharmaceutical University. And this study, Dr. Chifumi Kiyama joined this study when she was a medical student. She is now working as a physician in the hospital. Thank you for attention.
Thank you very much, Dr. Sakata. Really interesting, very surprising results. So let's start discussion. Do you have any? Oh.
Yeah, that was an interesting finding. I'm wondering, does that give the zebrafish a faster response so they can get away from prey? Is that the advantage, do you think? What's the advantage of having that physiology?
Yeah.
Do you think it gives a faster response time? So that's why I can never catch a zebrafish when I put my hand in the tank.
Yeah, we can catch. It's advantage of zebrafish, right? Yeah. Yeah, the main advantage. And so as I show you, we can easily knock out we can use the transient line and knock out, and also easily knock out line can be generated. This is advantage. And assay is also easy like this. We can just see under the microscope and just tap, and we can just easily analyze the movement movement of the fish. Yeah, okay.
So if there are no sodium channels, how can you have presynaptic depolarization?
Presynaptic.
So the vesicles are still released, yeah, from the presynaptic nerve terminals to cause muscle contraction.
Yeah.
So the presynaptic motor neuron, there's no action potential in the motor neuron, right?
Action potential generated in motor neuron. and acetylcholine is released.
I
And
see.
it
So-
gets open.
So in the knockout fish,
Okay.
the motor neuron can still fire
Yes,
action
fire,
potentials.
fire,
It's
of course.
because it's a different type of
Yes,
sodium
yes, right.
channel.
Yes, right.
It's the muscle that does not.
Yes. NAV1.4 is NAV1.4 is a muscle
Specific.
Yeah, muscle specific.
Okay, I got it. All right, great. Thank you.
Okay, time is up now. So thank you very much, Dr. Sakata.